For this episode of OMG OMx, you'll be hearing from Joey Latham, and I hope you'll feel as empowered by him as I have in learning about his journey. Hi there, Joey. Thank you so much for joining us on OMG OMx. Our viewers know how this goes, so why don't you start today by telling us your story?
Thanks, Kate. Thanks for having me on the podcast. I've enjoyed watching the episodes, and I'm glad to actually be involved. For my story, I grew up in a small rural town in East Texas and never planned on going into science or pharmaceuticals or anything like that. You pretty much either worked on the land somehow or you joined the military. So I really had no idea like what science or what any types of degrees in science could lead towards. I just knew from an early age that I was interested in science and was good in math and science growing up, and it was just kind of a circuitous route that I took where I just kind of made my way on my own to discover new things like a scientist tends to do. So yeah, it's not a typical path forward. No one in my family was in a science role. My dad was a coal miner. My mom was a housewife. Uh, Pretty much all of my family is still in the same area where I grew up. I'm the only one that actually left in the small town where I am from. So yeah, I am the black sheep of the family, or depending on who you ask.
So why don't you tell me a little bit about how you got into undergrad, how you made your decision to move forward in chemistry?
Sure. So when I actually started my undergrad career, it was pretty much a shock because I was going from a town of about 1,000 people. And I think my first class at university, the classroom was about the same size as my entire town. And so it was genetics. I had taken enough like advanced level courses and stuff in high school that I had skipped like the early biology requirements. So I was taking a junior senior level class, genetics, and you go into a lecture hall, and like I said, there was probably 500 to 1,000 people in the lecture hall, and so it was quite a shock. Again, going from some place where everybody knew me and I knew everybody else, to not knowing anybody, being 10 hours away from home, not having family around. So it was quite a shock. I actually started as out as a physicist just because, again, not knowing where or how people with a science field or science track, what types of jobs or anything there were. It was just physicists were the most famous. You always hear, Isaac Newton or Einstein. You're like, oh, they're physicists. I'm smart. I'll be a physicist as well. And it was only after time, lots of time, unfortunately, as an undergrad, uh, perusing lots of different classes and going to different labs and stuff that, I kind of settled on the chemistry mathematics track. And so it was really just trial and error. I went, I think, from a physicist to a chemical engineer to mathematician, and then finally a dual degree of chemistry and mathematics.
That's fantastic. And that's just so empowering, hearing from your early journey to getting to that point. So let's fast forward a little bit. You did your postdoc at Vanderbilt University under Richard Caprioli before transitioning into pharma. Can you tell us what it was like to do some of these things and the major differences you saw between pharma and academia?
Yeah, so it was great. One, being at a university like Vanderbilt, where just excellent facilities. It's a small undergrad campus, but a huge medical school presence. So you're really working at the cutting edge of kind of, cutting edge research and, institutional knowledge and basic research. And so it was a great place to be. And then, of course, Richard is so well known in the field, and just huge lab to join. I think at the time, Richard had something, like 15 undergrads or 15 graduate students, and then like 10 or 12 postdocs and research faculty. So, there are lots of people to learn from, and just a great overall experience. Um, the transition, from academia to pharma, and I talk to a lot of people that I hire about this transition because it is at times a very difficult transition, especially if you're fresh out of graduate school or you're a postdoc transitioning into the pharma industry. In academia, obviously you're, especially depending on where you're at in your career, you're writing a lot more, grants, trying to get funding, you're going to presentations, you're writing journal articles, a lot of things like that. The timelines are more drawn out, especially if you're on like a funding cycle, an R01 funding cycle. You're writing things or pretty much doing experiments to get your next R01. And that's probably the biggest transition going from academia to pharma. Is pharma, the timelines are much shorter. You have experiments where you need data returned out very fast. You need answers quickly, and again, it could be the answer you're hoping for, or it could be a negative result. I mean, the main thing is having the clear and constant communication back and forth with the people that you're working with. And so, the biggest transition, as I tell again, most of the people I hire, the timelines. You're under pressure to get things turned around very quickly, and just being able to open and honestly communicate those results with people because they don't care if the experiment didn't work. They just need to know the experiment didn't work, and they can't wait months to find that out. It's an accelerated, time frame that we have to work under, because time is money. And obviously, if you're in pharma, it is at the bottom line. It's a financially driven market, and you have to produce results.
So let's stick with that. Can you tell me why drug imaging is so important in pharma, and what are you looking for, and what's the information that's helping you make decisions?
Yeah. So for imaging, and specifically for MALDI imaging, it's important on multiple levels. I mean, for 20 years now, people have been using MALDI imaging mass spec to do to look for the drug distribution of potential pharmaceutical candidates. So it makes it much easier that you don't have to label the compound with some fluorophore or radio tracer. You're just doing native. You're doing mass spec, and you're looking at the drug or its metabolites in its unadulterated form. And so you're getting quick drug distribution where the drug is going in either the animal model or in preclinical tissues that you're looking at, or even sometimes clinical tissues. And so that's super important, and that's been around for like I said, over probably 20 years now. Pharma has been quick to take that part of the imaging aspect and bring it into the industry. Over the past, I'd say 4 or 5 years, you've had the advancements in instrumentation, the advancements in software, and some advancements in some of the ancillary and peripheral equipment that we use and the techniques that we use for imaging that have made it more robust, more precise, and have expanded the envelope of things that we're can look for. And one of the things that's helped with that, Ambergen, with their MALDI IHC probes. Now you can actually use the same, you have the same instrumentation, the same processing methods that you were using previously just to look for a drug or metabolite. And now you can expand that out to not only look for the drug and metabolite, but also, are you impacting the potential, protein changes that you're interested in, or the targets that you're hoping that your drug, actually impinges upon or it binds with. And so those things have opened up probably in the past 5 to 6 years, where not only we're starting to move beyond just using it for drug and metabolite imaging. We're looking for protein targets of interest, and now even more so to where you're actually doing really discovery level research, where you're actually using the imaging more along the lines of how you would do global proteomics or differential proteomics and transcriptomics, where you're literally just looking what types of biological or network changes do I see in either a genetic mouse model or a pharmacologically induced mouse model, and then what does my drug do in those instances. And so we've gone from looking for one analyte or one drug all the way to now screening and looking for these global changes in tissue that just 20 years ago there's no way you'd ever think that this was possible.
So, do the mouse models that you're talking about actually recapitulate the diseases that you're looking into? And I know PTC Bio has a really unique mission, which we'll come back to, but maybe you can comment on how it works going from mouse to human.
Okay. Well, I still have to work with pharmacologists all the time, so I will say that their mouse models do help us out. But yeah, as far as mouse models recapitulating human disease, for the most part, no. I think most preclinical in vivo models, they're tools, and that's how you have to think of them. They're a tool where hopefully, as a scientist, you have a hypothesis, you test the hypothesis, and that's just one mode of us doing that. Yes, we're not going to solve Parkinson's disease from an animal model. Uh, will we find out? Are there certain biological processes that happen in that disease? Do our drugs impinge upon that process? Yes, we can use animal models for that, but as far as fully recapitulating a human disease, no. But again, this is science. We have to use what we can, and just we have to make sure that we know that the answers we're getting, we're actually probing the correct questions, and so we have to just make sure that we're using those things as best as we can.
So, why don't you now tell me a little bit about PTC Bio, its mission, and maybe what drew you to working with them?
Sure. PTC Therapeutics. So we've been around for almost 25 years, and it's primarily a rare disease company that's focused especially in pediatric diseases. And so we work all across the world. Um, we work in a lot of places that traditionally big pharma doesn't like to go, because again, if you're working in rare disease, again, as we said earlier, pharma is a financial pursuit. And so if you're working in diseases where 1,000 to 2,000 patients around the world, yes, you can develop therapies for it, but the return on that investment is going to be quite small. It's not heart disease or cancer or anything like that, where you have millions and millions of people. That's those pursuits are still great to undertake those, but that's not our mission. Our mission is rare disease, pediatric disease, and it's a great mission to have. It gives you something to where, yes, you definitely want to come into work every day to work towards that mission, but it also means it's extremely difficult. It's hard. There's not a lot of basic research in some of these diseases. You don't have these big biorepositories of samples that you can interrogate, and so it really makes our own mission here in my group, the multi-omics aspect, it makes it to where it's that much more crucial, because you have a limited number of samples. For pediatric patients, you have small sample sizes, small sample volumes as well. And so you really want to get as much information out of those out of those samples as possible, because you're dealing, these are sick kids. They're it's you definitely don't want to waste anything. And so we have to make sure that, like, by whatever means we can, we get as much information from the that clinical population as possible.
Do you have any estimate or example of how many experiments you can do on one tissue section?
Oh yeah, I mean, for like all the time. So we routinely in my group, from one tissue, we'll run a global transcriptomics profile. We'll run a global proteomics profile. We'll run both metabolomics and lipidomics. We'll do drug quant. We have certain quantitative assays that we need to look for to make sure that we have, if it's in a, we have target engagement. So from one sample, especially with tissue, from one sample, we'll run 10 to 12 different assays. And so and again, a lot of those large data sets. And so, it takes quite a bit to actually now go through all of that data. But yeah, we try, again, we really want to get as much data out of these samples as possible, because again, you know, not only are these disease populations small, it also means that, back to your previous question about animal models, no one's developing an animal model for a disease to where humans there's only 1 to 2,000 humans that actually have the disease. So there's not a lot of preclinical information that is gleaned from animal models in these diseases. It's very rare that you have any preclinical data, at least in animals.
I know you have a strong commitment to mentorship within your group. So I think there's two sides here. There's an emotional load, and then there's empowerment. Maybe you can
Right.
tell us a little bit about both sides.
Sure. I mean, the emotional load, as we said earlier, we're working in rare diseases, pediatric diseases. So it's a lot of pressure, especially considering some of these diseases. There's no approved therapies, and so you're always striving to help the patient population. And so any failure that you have along the way, it can be quite debilitating because you take this very, very seriously. So there's that aspect of the emotional load. And there's also the emotional load of just, especially for younger career scientists, you know, first time in where we're at in the Bay Area, first time out of their graduate program or their undergraduate program, first jobs, and so everything is new and they're in a completely different environment. So there's also that emotional aspect of just being in a new challenge and being away from what was probably their core help group. And so trying to be with them in the lab as much as possible, having one-on-ones with them, just making sure that, forget about the work for a second, how are you doing in your own life? It's one of those things that, as I've gotten older and progressed in my career, the role of manager becomes as much about helping not manage, but almost as a therapist, where you're wanting to make sure that they're happy, because you want to make sure that when people come to work, that they're happy, that the environment that they're in is happy. And you could have the greatest mission in the world. And, I think we do. We're trying to help, like I said, sick kids. But if you can't come to work and be happy, then you can have the greatest mission and it's not going to be, beneficial for you. And so it's trying to make sure that you have a good group, that people get along, that you check in, that you always have open lines of communication, you're honest with them, and, just being a friend, and, making sure that they know that, like, they have somebody that they can talk to, and that if there's things that are wrong, either at work or in life, that they can bring those up. And it's something that everyone has to go through. And so you just have to be empathetic and make sure that they know that they can, like, rely on you and trust you.
Joey, you're really selling this. And I have to say, do you have any job openings? Because you never know.
Maybe, yes.
Just kidding.
We're
Just
all
kidding.
thinking about it. Then the empowering part, you know, to me, I'm really honest with my employees. I don't expect every employee to work for me for 20 years or work for, this company for 20 years. I expect milestones for them that just again, based on experience, this is kind of where you're going to have a decision moment in your life. Like, is this where, is this where you want to stick here? Do you want to transition to a new job? Do you want to rethink your entire career? I mean, because that's happened before. And it's making sure that whatever they want when they get to that point, I've helped them either way. And so whether that means, classes externally, whether that means like going through some of the things that they would do in senior management, making sure that they're ready. You just want to make sure that, again, they're scientists. They continue to learn throughout their career. And so you just, you always look for opportunities to make sure that you're helping them along the way, and then pointing out things that, that they don't know yet, that to let them know, are they interested in those things? And then if they are, making sure that they have the opportunities that they can. And whatever that is for them, I support it. So I want to make sure that, again, as a whole in life, that they're happy. And that doesn't mean that they have to stay here and work for me. But if whatever that career path is, I want to make sure they're prepared for it. And I will help them in whatever way I can. And sometimes that's networking, and sometimes, it's other things. But yeah, the main thing is I want them to be successful, happy people.
I really think you have a superpower here. And, I think this can be a really rare skill. So, again, I applaud you. I think it's really fantastic how much you take care of your team.
Oh, well, thank you.
Let me ask you one more question here, because we've talked before, and I know that
Yeah,
you have a really strong feeling and philosophy around DEI as well, and helping
yeah,
to empower groups that maybe aren't as well represented. You want to say anything on that?
yeah. I mean, I think it's super important. Again, going back to where I grew up. So again, growing up in a small rural town, I didn't have access to a lot of things, and I know a lot of people that grow up that way. I mean, I took math classes on a computer, and then I took physics in high school on a satellite TV with a phone where I would call in for questions. I mean, so I understand that, not everyone grows up with access to everything, and I want to make sure that no matter where you come from in life, that I can, like, that someone's there to try and help you along. And I also think it's beneficial. I think that, like, we all come with different like from we all come from different paths, and along that way, we all bring things along with us, and, the good and the bad. But for us, hopefully, we've learned from the bad things, and now they're actually even better, and there are more good things for us. And so I think the more people you can get with different paths, it just it starts to open up your mind to other views and to other ways to do things. And because we're, again, we have a goal, we're all striving towards that one goal. But the more people that you have with different skill sets, the more likely you are to achieve it. I mean, we don't have a pharmaceutical company made up of entirely medicinal chemists. That's, that you make a lot of compounds to test, but, who's going to test them? We have different, fields in science for reasons, because we all bring our own specialties, and we come together for a common goal. And it's kind of the same thing with hiring and having DEI. You want the diversity, you want the equity, and you want everyone to have a say. And I mean, I think it's the way everyone hopefully would want, to do business. But yeah, I fully support it. I want people who have their own specialties, have their own career path, can challenge me, can challenge conventional thinking, but at the same time, be open to other people's thoughts and their thinking as well.
That's just being a fantastic human, and thank you again for sharing that with us.
No, of course.
So maybe we could just end with, do you have any other fun facts or anything else that you want to leave people with today?
Uh, I have lots of fun facts. You can ask my team all the time. No, they ask me quite a few times if they're actually true, and for most part, they are. Uh, you know, again, I had no idea. Like I said, my dad was a coal miner, my mom was a housewife. I had no idea what I was good at, where this career was going to take me. I think a lot of it, is you just need to be open, open and inquisitive, and it's interesting where you get with that mentality. And then again, like I tell the people that I work with, have fun. I mean, don't take this home work. Don't take this work home with you. Yeah, it's fine to think about it, and it's great. Sometimes you'll be doing something, and all of a sudden you have a light bulb moment. That's great. But in order to have that light bulb moment, sometimes you have to get away from the job, and you have to get away from the work, and you have to be in. You have to have some sort of peace of mind. And so if that's playing a sport, if that's hiking, if that's surfing, if it's doing anything, I, I have people that are sewers, that are gamers, that are whatever it is for you that allows you to kind of unplug and disengage for a while, where that meditation, whatever. I think it's something that you need, and uh, who knows? There's no telling what you may discover about yourself, and potentially what you're working on, if you actually give your brain the rest and the time that it needs to find some of those things.
Yeah, that's fantastic. I've seen some recent studies too that say your productivity are increased when you get that break. So it's nice that you can implement that and encourage that.
Yes.
All right. Well, thank you so much, Joey. It's been a pleasure as always chatting with you. And thanks for being on OMG OMx today.
Thanks, Kate. It was great.
Listening to Joey today and being able to talk with him has reminded me of a quote by Albert Einstein, who once said, "I have no special talent. I'm only passionately curious." And so I hope you can take that with you today and feel more invigorated from what he said to us.